Number 21. And in this podcast, we're going to talk about the origin of the nucleus and how it is that you can pack all that DNA into a small space, the nucleus. Okay, so the first people who are looking at this looked at it with electron microscopy. Electron microscopes allow you to see things much, much smaller than your standard light microscope that you would see in a principal's lab, for example. And they could see things that looked like DNA, and they looked at a virus genome. So this is not inside a nucleus, but just a naked piece of DNA inside a virus. And they could see that it looked like in some pictures that it was like beads on a string. So you see these individual beads and then sort of like string connecting these beads. And then if they extracted for protein, get rid of all the protein, you see what just looks like the string and not the beads. So maybe the beads are made of protein. Other preparations, you get single beads with a little tiny string hanging off of them, 2 beads and 2 bits of strings connecting them, 3, 4, etc. If you separate those in a centrifuge, that's not how you spell centrifuge, it's centra, R-I, fuge, then you'll have things separating according to size, and you have a big particle. This is using UV light to see where things are in the centrifuge. So you have a big particle that corresponds to 4 of these beads, and then 3 of these beads, and then 2 of these beads, and then 1 of these beads. You could separate those out, and then you could take each one of these that are separated by the centrifuge and run them out on an electrophoresis gel, just like before we saw the electrophoresis gel when we looked at different kinds of RNA, and you stain for DNA against known sizes of DNA. This is called a size ladder over here. So this is 200 base pairs, 400 base pairs, 600 base pairs, and you can get an idea of how much DNA is wrapped around one bead, and it looks like about 200 base pairs. In later pictures of electron microscopes where they looked at different compaction of DNA and DNA under different situations, so not stripped of all the proteins, they get this sort of structure, this sort of model, where the protein is the circular part, the bead, and then the DNA is wrapped around those beads, and about 200 base pairs of DNA is wrapped around each bead. And then the beads on a string can get wound up into higher order shapes of winding. So most of you guys don't remember this, but phones used to have cords on them. Your parents would remember this. Or in an office that still has a landline phone, there's a cord attached to it, and the cord always gets tangled. And there's coiling, and then there's what's called supercoiling, when the coil starts to coil on itself. And that's what happens in your DNA too. So we're going to watch a little animation here. So you can see that.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope: chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay, so returning to this, it's important to know that when you see chromosomes, like in a diagram on mitosis or meiosis, that is when DNA is the most compact, and that's when the cell is moving around that DNA. When the DNA is all wrapped up and really compact like that, like you can see it as a chromosome, it's very hard for enzymes to get at that DNA in order to read it, in order for transcription to happen, and then after transcription, translation. So when DNA is tightly packed like that as a visible chromosome, there's not a lot of transcription going on. And when it's diffuse as chromatin, it's still associated with the histones, but now you could unwrap a histone and transcribe that DNA and then rewrap it. We know that if you chemically modify these proteins, these histones, and you can chemically modify them just by things in your diet or things that you're exposed to environmentally, it changes the wrapping of the DNA and it changes the ability for transcription enzymes to get at that DNA. And so it can affect the likelihood that you're going to transcribe a certain section of DNA. And this can lead to all kinds of problems and differences, like it can lead to obesity, it can lead to hypertension through modification of histones on which the DNA is wrapped, and that changes how accessible that DNA is. So histones are a way for a cell to take 6 feet of DNA molecule, wrap it all up, and put it in a nucleus. And that's an important advantage in eukaryotes. Bacteria don't have histones, and so their so-called DNA is naked, and they don't have protein associated with it. Also, their DNA is in a circular form. Archaeans, which are another kind of prokaryotes, do have histones. And this is an amino acid alignment where you take the sequence of histone proteins and you line them up with the sequence of lots of other histone proteins from different organisms. And the point of this is to show you that the sequence of those proteins is very highly conserved across a lot of evolutionary distance. Here's humans here and its sequence of histone proteins, and here is a palm tree or corn, for example, and you can see that compared to the human sequence, the histone from corn is almost the same. So usually that's a signal. If the sequence has not changed through that much evolution, that means that that sequence is probably really vital to the survival of that organism. And if you change it, you can change the survival of that organism. Maybe that organism won't survive. So when you see high conservation like this, it means usually that protein is essential for survival. So you don't see bacteria on this because bacteria don't have histones. And the fact that bacteria don't have histones is one of the characteristics that we use to say how we are related to prokaryotes. So archaeans, because they have histones, some people think that makes it more likely that eukaryotes came from archaeans. But as I said in the last podcast, there are other characteristics that are closer to bacteria, like the DNA inside mitochondria and chloroplasts. So we have characteristics that put us closer to archaeans and characteristics that put us closer to bacteria.